everyone. My name's Louise. I am the Territory Manager at Millennium Science for Instruments. I'm based here in Adelaide, and I'm here with Andrea from the lovely Waite Institute here in Adelaide. And she is a master's candidate in wine microbiology at the University of Adelaide. And she works within the CRCP advancement of Australian lifestyle wines. She's a student under Christian Varelera, and her research focuses on improving microbial stability and sensory quality in dealcoholized wines. It's a bit of a tongue twister. And so she's going to present some research that she's been doing using the Agilent Biotech log phase 600 instrument that we installed last year. So I'll hand it over to Andrea.
Thank you for the introduction. I'm going to share my screen. We practiced before. See how it goes. Okay, there we go. Can you see my screen? So, yeah. Is that okay? Can you confirm?
Yes, we can see.
Amazing.
Perfect.
Wow, beautiful. Okay. Well, thank you for the introduction. As it was mentioned, my name is Andrea Bustos, and I'm a master's student in the University of Adelaide, part of the big CRCP NOLO project, and we are trying to make non-alcoholic wines better. So today I will be sharing with you some of the research we have been doing on this area. And before, I would like to start with sharing a phrase with you guys. My mind wants that experience and wants that ritual, but my body is like, I don't really need the alcohol for that. And this makes sense when we look at the effects that alcohol has on the human health, right? So we all know that alcohol consumption have effects in short terms, like unconsciousness, hangover, intoxication, injuries, and then also there are linked diseases with long-term use of alcohol, such as certain types of cancer, cirrhosis, cardiovascular diseases, and brain damages. But it's also linked to mental health with like depression and anxiety. So these effects have been showing over the last 10 years decrease in worldwide consumption over the years. And this is also maybe associated with the no and low market value that in the same period of time have been increasing over the years. So what do we mean when I say NOLO wines? So the OIV defines the alkalized wine as a beverage that has undergone the fermentation of fresh grapes. And after the fermentation, it has the alcohol has been removed deliberately. There are two main categories, non-alcoholic wine and low alcohol wine. The exact limits can vary depending on the legislation of each country. And for example, here in Australia, we have two categories for these NOLO wines. These are below 0.5% of alcohol are declared as non-alcohol, and they don't need a statement in the label. And then we have a low alcohol category, which is between 0.5 and 1.15% of alcohol. So in short, these beverages are still real wine by definition, but they have the alcohol been removed by a process. So to make sure that we are all in the same page, I'm going to talk about the winemaking process very, very briefly. So if I have any winemaker here, this is not going to give you much detail. But in short, the winemaking process includes the fermentation process, where the yeast will convert sugars that are in the grapes. These are glucose and fructose. Yeast convert this sugar into ethanol, CO2, and aroma, which are going to give the appealing flavor and aroma of wine. Usually wines after fermentation undergo a aging process where it gives more complexity to the sensory characteristics, and also a filtering process to remove all the bigger particles. But also, and in this case, NOLO wines can undergo a dealcoholization process. After this, both types, normal wines, full-strength wines, or dealcoholized wines, need the addition of antimicrobials or preservatives. And usually in winemaking, the most commonly used antimicrobial is sulfur dioxide. This is an antimicrobial that is used in many industries, and in the winemaking process is also used before fermentation when grapes are harvested. And it not only has antimicrobial properties, but it's also a very powerful antioxidant. So it has both properties, antimicrobial and also antioxidants. And then wine can be bottled. But how do we dealcoholize wine? How do we reduce the alcohol content in wine? So there are three main stages where we can actually do that. So these are before, during, and after fermentation. So before fermentation, the goal is to reduce the sugar concentration in the grape berries. This can be done by viticultural practices. And then during fermentation, the aim is to reduce the ethanol production, and winemakers can use different strains of microorganisms, or also they can stop the fermentation before it's complete. Sorry. But these two practices only allow to reduce the ethanol concentration by 1% or so. So the only option to completely remove alcohol is after fermentation, which uses the physical properties of ethanol. So we can remove alcohol by membrane filtration or by vacuum distillation. And in that sense, we can obtain a wine that has around 0.5% of alcohol by volume. So this is how we can remove alcohol, but NOLO wines have a few challenges if you have tried them. So one of them is the microbial stability, and the other one is the loss of flavor and aroma. Microbial stability is a challenge because when we remove alcohol, alcohol works as a natural preservative in wines, in beverages in general. And winemakers also add sugars to improve mouthfeel and sensory characteristics. So this represents a perfect environment for microorganisms to grow and potentially spoil or ruin the products. And then also when we remove alcohol, we are not getting removing the spirit of wine, but we are also removing part of the important flavor. Today I'm only going to talk about the microbial stability part of the things. Otherwise it'll be maybe a little bit longer to talk. But for this part of the research of microbial stability, we took samples from two different wineries, a Chardonnay and a Sauvignon Blanc, at different winemaking or dealcoholization steps. We have a base wine, which is the full strength wine. Then we have a sample after dealcoholization. In the case of winery 2, we have the alcoholized wine after addition of sugars, as I mentioned before, and then after the process of filtration, when they remove the bigger particles. Then this wine stayed in a tank for a period of 2 weeks or so before bottling. And before bottling, it's also filtered, and winemakers add antimicrobials. In this case, they added DMDC. And then we have a final product, 0% alcohol. So what we did with all these samples, From each bottle, we took a sample of wine and we extracted DNA to evaluate the population, the microbial populations that were present in each sample. This is the DNA contained in each sample, so it's not microbes. We are looking into the DNA exactly. And then we also plated these samples in agar plates to isolate and count the microbial load present in each sample. The difference, I was talking with a friend, and the main difference, a friend that is a chef, she's nothing to do with science, and she was like, what is the difference between these two? And that made me think, how can I explain this to somebody that is a chef? And I think the main difference is that here we can see either alive or dead microorganisms. So because we're looking into DNA, whatever that is dead but left the DNA there, we can see that. But here we can only see things, I mean, microorganisms, sorry, that are alive and can actually reproduce when we put them in a plate. So we did that. We counted and estimated the microbial load that was present in each wine sample, and we took isolates that we then sent to sequence so we can determine the identity of each isolate, and then we also characterize them under different conditions. So here I will explain how we proceed with the wine samples. Each wine sample, was taken in duplicates. The first bottle, we opened it at time point 0. We took a sample for analysis, and then we closed the bottle and put it back in the fridge. And after 6 months, we opened it again and took a sample for analysis. For the second bottle, we kept it closed in the fridge for a period of time, and only after 6 months we took that bottle, open it, and take a sample for analysis. When I say open the bottle, I mean in sterile conditions. So it wasn't in the bench; it was in the sterile hood. So keep this nomenclature in mind, because the next slide will show the results for these experiments. So here's an example of how the plates look like after microbial growth. We plate 50 microliters, a small amount of wine on these agar plates, and then let them grow. And then we count the concentration of colony forming units. This is what you can see here for winery 1, for winery 2 in purple. And you can see the time point here, time point 0 after 6 months. And this was the bottle that was open at time point 0, and then after 6 months of storage. The first thing that I'm going to show you is the step of filtration. At this point, there was a filtration step to remove big particles that can be a microorganism as well. Here in winery 2, there was long storage periods, and after additions of antimicrobials, in this case, TMDC. What you can see here, the darker the color means more concentration of cells, and you can see that after a filtration step, there is a decrease in the numbers, but then after a long storage period, the numbers increase, and then when you add the MDC, the numbers decrease once again. And this, you can see that for all the time point of sampling. So key takeaway for winemakers here would be like, make sure once you dealcoholize your wine, make sure that the period before bottling and adding the MDC or whatever antimicrobial you use, it's as quick as possible, because at this point is where your wine can get spoiled by the microorganisms growing there. So make sure you are fast when you're making NOLO wine. What did we find in these samples? In winery 1 on the left, winery 2 on the right. The first thing that you can see is the orange color, which is unsurprisingly Saccharomyces. I have to mention this is yeast populations. you remember previous slides, I talked about the experiment of evaluating, what was the name? Evaluating microbial population. So this is when we took samples of DNA within each bottle. And what you can see here is the DNA expression of the microbes for each sample. So in orange, you can see Saccharomyces is well represented in both wineries, both locations, and every single dealcoholization step has Saccharomyces. But then you can see that the dealcoholization process also shifts the microbial population. You can see in light blue, Kurtzmanniella. Kurtzmanniella is a yeast that can ferment, so that represents a risk for NOLO wines, because once you remove the alcohol, if you have the presence of a yeast that can ferment, it's going to produce ethanol. And that is not going to be non-alcoholic wine anymore. Then here in winery 2, you can see purple lavender color. This is weaker Saccharomyces. This is a non-fermentative yeast, but it can produce off flavors. So that is also something that can produce spoilage in your wine. And then here you can see the apricot color, which is Rhodotorula. Rhodotorula is a yeast that have been presented as a potential pathogen recently, so that can represent a risk for safety consumption for the consumers. Talking about numbers, so far we have isolated more than 800 microorganisms. More than 500 were yeast, more than 200 were bacteria. We are having the phenotyping underway, and in total we have phenotyped 144 yeast isolates, and we have characterized these isolates under 3 different treatments. First, a control, and then we tested them in ethanol concentrations, which is a natural antimicrobial. Then also we tested them under different concentrations of sulfur dioxide, and also DMDC, which is the antimicrobial I was talking about previously. So phenotyping, what is phenotyping? Phenotyping essentially is a way to characterize microorganisms, in this case, under different growth conditions. So what we are looking here is a microbial growth curve. You can see that the more microorganisms, the denser the media gets, and we can measure that as the optical density, and that's why we're using the log phase 600. We're measuring different conditions. So first is our control condition, which was real dealcoholized wine. This was a Chardonnay with the addition of glucose and fructose. And we also tested the 3 different treatments: ethanol, sulfur dioxide, and DMDC, all of them in different concentrations. And here you can see a real growth curve of Saccharomyces cerevisiae, EC1118. EC1118 is a widely used yeast in the winemaking industry. And this is the growth curve under control conditions. So under Chardonnay with 15 grams per liter of glucose fructose. But what happens if we add antimicrobials? They're not so happy. the growth curve is not as high as the one in control conditions. So from this growth curve, we can have different kinetic parameters, and the one that I am going to be looking into is the area under the curve, which summarizes all these kinetic parameters in only one data point. And you can see that if you compare the area under the curve under control conditions versus an antimicrobial, you can see that it is much higher without the antimicrobial. So for this, we use the log phase 600, which is a high throughput microplate reader which is designed for microbial growth studies. It allows us to record optical density in real time under control conditions such as temperature or shaking, and it's giving us a very detailed growth curve data that we are processing and using for our research. The thing that I love of the log phase is that we can read 4 plates at a time, and we don't have to be in the lab to measure because it does it for yourself. So you just put the plates and see you later. 3 days after, I can come and collect the data. It also has a very friendly app, so you can just adjust the conditions that you require. You can set the temperature if you want a gradient to prevent condensation on the lid, so you can have like more precise data, not blurry data points or weird data with the condensation on the lid because the reader wouldn't read as well. Then you can also have shaking, which gives more real scenario because the cells are not getting in the bottom of the well. And so for my experiments, we were measuring at 600 nanometers. The log phase 600 obviously only measures at 600, the name says it. I ran my experiments for 3 days at an incubation temperature of 30 degrees with shaking the whole time. And after that, we get the raw data, the measure of the optical density. And we process this data using RStudio with a package called Growth Curver. So this is how the data looks like after we process it. Here you can see 96 growth curves. In total, I have phenotype, as I mentioned, 144 isolates in different conditions, treatments. It gives us a total of around 3,000 curves, which is quite a lot if you want to compare and analyze them individually. So with the growth curve package, we can summarize all this data, all these almost 3,000 growth curves in only one plot, which is quite handy when you need to see or compare the different treatments. So what is this plot showing us? Here you can see in this plot all the 3,000 growth curves all together in only one plot. You can see in the y-axis the microbial growth, and in the x-axis you can see the different treatments we have. The first bar plot is showing you the control media. This is the Chardonnay with sugar. The black dots represent the area under the curve of each isolate. And you can see in red the positive control, which was EC1118. And here you can see the different treatments. So ethanol and different concentrations. The more concentration of ethanol, the smaller was the area under the curve. So in other words, that means that if we remove the alcohol, there is a potential for microbes to grow more, I don't know, easily or more. That is represented in the area under the curve. So the less ethanol, they can grow better. And a similar trend we can see when we add different increments of sulfur dioxide. So you can see that the more sulfur dioxide, they grow lesser. And the only antimicrobial that worked effectively was the MDC. So nothing was growing at this point at 2 different concentrations. So the big issue here would be that the MDC is so far the only antimicrobial that we have that it's effective and against all the single isolates that we have from NOLO wines. So it's only a matter of one single isolate that creates or develops resistance, or legislation in different countries can change and then they can ban the MDC. So we need to find different alternatives for antimicrobials. And this is something that we have been doing in the lab. So we are evaluating different potential antimicrobials. So far, we have analyzed 4 products. These are chitosan, tannins, fumaric acid, and PBI, PBP. We tested these antimicrobials in not antimicrobials products in 2 different yeasts, Saccharomyces cerevisiae and Brettanomyces bruxellensis, and 2 different bacteria, Acetobacter pastorianus and Gluconobacter oxidans, which are common wine spoilage. Specifically, Saccharomyces cerevisiae is a spoilage in the NOLO wine context because it can re-ferment the NOLO wine. And I'm going to show you the results. So briefly, none of these products tested can effectively control Saccharomyces cerevisiae. Only PBI PBP showed minimal inhibition without sugar, which is pretty unfair if you think that the yeast doesn't have any carbon source to survive. And then chitosan was effective to control Brettanomyces bruxellensis. But yeah, we still have a lot of work to do in this regard, trying to find alternatives to antimicrobials. And this is, I'm reaching the end of my presentation. So as a summary, I can say that the abundance and diversity of microorganisms are affected by time and NOLO winemaking practices. The population structure is dominated by Saccharomyces. The microbial growth decreases with stress agents, but some microorganisms are more resilient, and the absence of alcohol can allow microbial growth. DMDC is the only antimicrobial that works effectively in NOLO conditions, and more research is needed to find out antimicrobial alternatives. So before finishing, I would like to acknowledge the CRCP NOLO project, particularly the bioengineering pillar. My teamwork, my team in the lab, they are amazing and very supportive. And obviously my industry partners and Agilent, I didn't put your logo here, but thank you so much for the opportunity to present my research. And I will be happy to answer questions if you have any.
Thank you. That was wonderful.
Thank you.
I'll start off with a question, which I thought of while I was listening. So have you looked at any red wine, like NOLO red wines yet? Or is there any differences? Or would you perceive there would be any differences considering that red wine has more tannins in it?
That is a good question, and we are planning on analyzing a red wine. We haven't done it yet, but it's on the agenda. So we will have that data soon. And yeah, I think it's going to be different. It will be different because red wine has a lot of tannins and other type of molecules that can contribute to like preserve better the wine.
Yes, that's right.
I think it's going to be different. I'm expecting to see different things.
Okay, great.
Yeah.
I'm just checking with David. Was there anyone else that had a question? You can either put up your hand or put it in the chat.
Yeah, I don't see any hands up or questions in the chat at the moment.
Well, if anyone has any questions after the presentation, we will be, obviously this presentation was recorded, so we will be sending this out to everyone who has attended, and those who haven't attended. Oh, here we go. Here's a question. It says, do you think it's possible to make coffee NOLO wine?
Coffee NOLO wine. I
know
mean, you can do
some
whatever
coffee
you want.
technology
If
today.
you have the materials and the time, you can do whatever you want. Like, I don't know, how do you imagine this product? But for sure, you can have coffee extraction to wine, but up to you if you like that. It's interesting, honestly, but I don't know if you can tell me a little bit more about the idea, because I'm not sure if I'm thinking what you think.
And another question from David Bean is are NOLO wines often pasteurized?
That's a very good question. No, usually in the wine making industry, they don't pasteurize wine. It's not used, but in beer, yes, they do like a flash pasteurization, and that makes things, I think, maybe a little bit easier. It's another tool to prevent microbial spoilage, but in wine they don't use it.
Interesting.
So usually sterilization, you can use cold sterilization with the MDC, which works really, really well. But as I said, like if it changes the legislation of any microbe develop resistance, that's it, right?
Yeah.
There's also the use of filtration, but yeah, no, pasteurization is not used.
Great. Thank you. Yes, as I was saying, we will be sending the presentation out. And here's another question. Other than tannins, do you think caffeine or other phenolic compounds can inhibit the microorganisms' growth?
I didn't think about caffeine as something that could prevent microbial growth. I'm gonna read about that, so that's something that I'm gonna take notes. Phenolic compounds. We've been testing tannins, but they didn't work so well. So it's something that we have to look at it. And they also work better with ethanol concentration. So yeah, that's all I know so far. So we have to keep looking into alternatives.
Yep.
But I'm gonna see if there's anything related to caffeine. That's very interesting.
Yes. Great. Well, I think if there's no other questions, we can wrap it up. No one else wants to ask a question. So I'd like to thank you, Andrea, for your presentation. And then, yeah, I'll see you soon.
See you soon. See you soon in the lab. Thank you so much. Bye.
Thanks, everyone. Have a great rest of the afternoon.
Thank you.
Bye.